Good evening. Very good evening. Good evening, Manoj. Good evening, Dr. Sridhar. Good evening. Very good evening. You are on mute.
Let me know when I have to share my title slide.
Yeah, sure. I think the organizers must be doing it, I think.
Okay.
I say I would let you know now. I'll ask.
दत्ता साहब गुड इवनिंग।
हाँ, गुड इवनिंग, गुड इवनिंग।
और बाकी लोग जुड़ ही रहे हैं।
अच्छा अच्छा,
दे आर या ज्वाइनिंग द मीटिंग।
Good evening, Satya sir. Good evening, Manoj sir.
Very good evening.
Good evening, everyone. Okay.
Good evening.
Good evening.
So,
మీటింగ్ జరుగుతుంది అమ్మా
on behalf of FIDR, I, Aishwarya Pandey, Knowledge Lead FIDR, welcome you all to this edition of Think Hatchery Talk series. FIDR is India's leading think tank. Today's talk is on growing significance of genomics in India. We are very happy and honored to have Dr. Sridhar Sivasubbu, Senior Principal Scientist, CSIR, Institute of Genomics and Integrative Biology India as our esteemed speaker. He would address us on growing significance of genomics in India. Now, we have participants from more than 6 states of India and from outside India. This includes professionals, bureaucrats, practitioners, and prominent civil society activists. I would now request Dr. Satya Bhushan Das of IIM Lucknow to introduce Dr. Sridhar and topic for all of us. Over to you, sir.
Professor Das, you're on mute. We can't hear you.
Okay, thanks. And it is my pleasure to welcome all of you for this webinar from IIM Lucknow also, Center for Marketing in Emerging Economics. Our genomics is now integral to any disease research and drug discovery due to its implications of genetic expression and human health. And genomics has scientifically proven its ability to prevent, manage, and treat diseases. Precision medicine is gradually replacing conventional treatment methods in the healthcare system. And during the past few years, this transition has been sped up by extraordinary clinical qualities, the qualities and infrastructure. Detection of serious disorders like cancer is now made possible by the development of whole genome sequencing as a valuable technique. The genomics industry is expanding due to factors such as increased demand of gene therapy, customized medicine, drug development, and an increase in cancer incidence worldwide. Furthermore, a statistic says the global genomics market was worth USD to 24.16 billion in 2021 and is expected to increase at a compound annual growth rate CAGR of 16.4%. It is very high between 2020 to 2030. and Asia Pacific is estimated to be the fastest growing segment over the forecast period due to increased demand of genomics application in diagnostics and the growing demand for novel therapeutic drugs to fight the increased incidence of diseases in the region. India holds a huge potential for dividing the whole genomics industry. The Genome India project was launched by the Department of Biotechnology in January 2020 to create a reference genome. The project intends to gather 10,000 genetic samples from Indian residents throughout the country. Precision health, rare genetic disorders, the mutation spectrum of genetics and complex diseases in the Indian population, genetic epidemiology of multifactorial lifestyle diseases and translational research are some of the areas of this project attention. With this, that means there is a significant potential of this genomics and precision medicine. And there are also, being a professor in marketing, there are also challenges of opportunities. Today, it is really, we have got a very good opportunity to listen from a distinguished senior principal scientist, Professor Sridhar Sivasubbu, who is now head of Business Development and Marketing Group, CSIR, Institute of Genomics and Integrative Biology, Delhi. And Professor Sridhar is a senior. It is my pleasure to introduce him because he's a very renowned personality. Most of us know he's a well-known name in the field of genomics. Dr. Sridhar is a senior principal scientist at the CSIR Institute of Genomics and Integrative Biology, Delhi, India. His research interests are focused on sequencing human genomic genomes for adoption and benefit in healthcare with applications in rare genetic disorders. He also develops geographies models for application in precision medicine in humans. His laboratory also explores non-coding RNA-mediated regulation of vascular development and diseases, wherein his group is credited with the discovery of a number of non-coding RNAs. His group also piloted and leads the genome sequencing-based surveillance of SARS-CoV-2 for India and Southeast Asia. Dr. Sridhar co-founded the Genomics for Understanding Rare Diseases India Alliance Network, popularly known as Guardian, which is one of the largest precision medicine ecosystem in India, working on rare genetic diseases. He also served as the Chief Scientific Officer of the Center for Genomic Application, a public-private partnership company established by IGIB. Dr. Sridhar completed his PhD from MS University, Tiruvallur, India, and postdoctoral research from CSIR Center for Cellular and Molecular Biology, India, and the University of Minnesota, USA, with specializations in genetic and genomics. His major research areas are human genomics and precision medicine, geographies models for application in precision medicine, non-coding RNA-mediated regulation of vascular development and diseases. He has around 150 publications, international renowned publications in his area of research, and 5 technologies commercialization, 27 molecular diagnostic assays. With this introduction, I don't want to linger more on his introduction. And now it is my pleasure to invite Professor Sridhar to address this gathering, and will be really benefited to listen from Professor Sridhar.
Thank you, Professor Dash and the organizing committee of FIDR and IIM Lucknow for the invitation. And thank you, Professor Das, for the elaborate introduction and to the field as well as my own profession. I see a number of candidates here, participants here. So let me not take too much time. I'll start the presentation but it's a pleasure for me to address this August gathering. With that, please allow me to share my slides.
Yes, sir. Yes, sir. You can share your slides.
1 minute. Okay. Is my slides visible to everybody? Can somebody just confirm it?
Yes, sir. Your slides are visible to us.
Okay. Okay. Thank you. Thank you once again to the organizers. For the next 40 minutes, what I'm going to do is take you through a journey that I am very passionate to have been part of, and something that I dreamt as a student, as a college student, that somehow I would be able to undertake, and I'm really happy that I could actually take it forward. With those words, so what I'm going to do today is talk about the not only the significance of genomics in India, which has been growing for the last decade plus, but I'm going to really focus upon how has genomics not only been as a tool that is exclusively used for a few individuals. Rather, I'm going to talk about how could you use this tool for a public health benefit, which is a much larger canvas for any one of us to fulfill. But before I do that, I just have a slide of to introduce where am I coming from, and how do I do this whole operations. So the CSIR Institute of Genomics and Integrative Biology is one of the 37 labs under the Council for Scientific and Industrial Research in India. And we were set up to translate concepts developed in basic biological research to commercial viable technologies for healthcare. We became in operation in 2002, and we have been in existence since then as a pioneer genomics institute in the country. We have a very modest scientific strength of about 42 scientists, but what we are really proud about is the depth of expertise in disease biology and translational research with a very unique interdisciplinary team. There are no two people in the institute with the same kind of expertise. So you can see how diverse we are in terms of our expertise in clinical as well as informatics and basic biology. With those words of introduction to my institute, let me quickly go back to the start of this millennia. For about 20 years, and about 6 countries in the world had put together this program to sequence human genomes. Now, human genome is nothing but reading the DNA that is contained in all our cells. Now, this DNA is actually about 3.3 billion bases. Imagine 3.3 billion data points packed into every cell that you have in your body. Okay, that's the amount of information. We are a walking, talking, moving set of data points in terms of billions of nucleotides. Okay, now reading this out was a humongous mammoth task, which started in the late 80s, and by 2003 we had a very decent draft, but it did cost an estimated $2.7 billion US. Okay, and in the screen are 3 gentlemen. The center one, most of you would recognize as the former President Bill Clinton of the United States, but the 2 gentlemen flanking him are scientists. One led the government program, and one led the private initiative. Okay, and now between Craig Venter and Francis Collin, they could manage to put together this first reference genome, which finally came to these 2 major conclusions. One was that human genomes have about 25,000 protein coding genes. Okay, I'll not go into the details of genes, but 25,000 building blocks that make up what you are in terms of proteins. But the downside or the bummer in this whole exercise was that, okay, 99.9% of the 3.3 billion bases are identical between 2 people. So the person you and your friend are identical by 99.9%. That means you're differing by only 0.1%. Okay, so the thought is the whole genomics revolves around this concept that if you are able to decode this small percentage of difference between people, that would explain why we look different, why we behave different, why do we have different diseases, why do you now respond differently to different diseases, and it has a whole new field of human clinical application that could be now figured out. So that's the whole science of the human genome. And this was one of the promises that was offered, that we would be able to make sense of this and use it commercially and socially for benefit of humankind, was the initial thought process that was put together. But like I said, it's all about me. It's a small difference in me and you that if you can figure out, you really know why we look different, why we behave different. So this led to this concept of human genome. So here is the first decade of the human genome after it was unraveled. So in your x-axis is the number of years, and the y-axis is the billions of base pairs that one could sequence, and the red thick line that goes right through the middle of your screen is nothing other than the cost of sequencing drastically dropping very rapidly. Okay, in fact, so rapidly that today you don't need billions of dollars to sequence one human genome. You could do that for as less as about $350. That's the current technology in offering. Remember, it costed, the initial genome did cost us close to $3 billion. So people like me, when we had graduated out, it was a dream to be part of any genome sequencing exercise. And we started our labs. I started my labs in about 2007, and by 2007 you could see labs were starting to do genomes. The first individual genomes are starting to appear from the reference genome. So I took up this opportunity, really leap forward, saying that could this be an opportunity for India to really scale up or leapfrog into the field of genomics? Is a question that we asked ourselves, but we did not even know the basic expertise to go about doing it. That was the challenge. So with this challenge, we started putting together expertise in my lab and my colleagues' lab, and that led us to actually sequencing the first personal human genome from India way back in 2008-2009. And as you can see here, this one single personal genome that we did at that landmark event today has laid the foundations to what we are seeing in genomics as a revolution in the country. And what did it tell us? It really taught us how to make sense of the genome. What does it mean in the clinic? How could you use this for deciphering what the disease the person carries, so on and so forth. But I'll give you examples of how to take this forward. But we didn't stop there. What we quickly did over the next few years was to realize that just by doing genomes in India, you'd not be able to make sense of what's going on. You need to do genomes around us. So the first genomes out of Sri Lanka, Malaysia, Philippines, so on and so forth were all done in our labs. So for two reasons. One was to demonstrate the scientific expertise that we had developed as commercial products. Okay, so the first genomes actually were done as commercial products for other countries way back in 2008-9. But more importantly, show a collaboration, a South-South collaboration that could happen, because this technology was a niche area technology, and the Western countries were literally not ready to part with it. So this demonstrated that one could develop such technologies, learn them, and be able to distribute them around us, so that we all be collectively able to answer a question. So today we have a large collection of genomes, not only from India. So we recently completed the 1029 genomes in India and published them. I'll talk about it. But Southeast Asian genomes, now we have a lot of programs going in the Middle East that we participate in. So you can see it's a rich diversity that we cover. But all this, what do you do with such a data, was a question that we really mulled over for a long time. And remember on December 8th, 2009, the first human genome in our country was announced at the parliament by the then Science and Technology Minister. But really the question that the common man, or rather my scientific paymasters, and as well as a lot of the scientific community was asking, why would you want to do something which is 50 lakh rupees expensive? So the cost of doing genome sequencing in 2009 was 50 lakh rupees per genome. And it was a very unsettling thought process. But on 9th December at 6 a.m., I got a call. I call it the Bhai calling. This person's last name is Bhai, and he's from a very remote place in Gujarat. And he calls me at 6 a.m. in the morning and telling me, मैं भाई बोल रहा हूँ, आपको पता नहीं आपने क्या किया है, but ये हमारे लिए ना आप देवता हो. I didn't understand the statement for a long time. I thought it was a prank call. But finally, when it made sense, was that he was calling me because he represented the world's largest family with a genetic disease. I didn't realize that a common man who reads only Gujarati newspapers got the message so clearly what genomics can impact him and his family and his community. And I was struggling to explain that to many other people in the learned community. But nevertheless, that led me and my colleague to Vinod Skaria to really look at it very seriously. And along with Bhai, we evolved this program. Okay, and we realized that genetic diseases are a hidden social burden in the country. Each patient with a rare genetic disease goes through 8 physician visits, 3 misdiagnoses, and 7 years to diagnosis. And there are not a few of them. There are 70 million people in India and 300 million people worldwide who suffer from such rare genetic diseases. So that was right there. Why do we need to do genomics? Could we boil this whole experience to the first year of life, if not the first few days of life? Could we do that? Was the question. That was the challenge. That was the goal that we wanted to do. Could we impact human lives? Could we have healthy babies being born by using this technology? Okay, that was the question that we really wanted to address. And why India? Large population, 1 billion+ people, diverse genetic ancestries, very stratified communities, but most importantly, you had close-knit family structures. Who would have thought that one of the largest families with this genetic disease came from Bhai's own family? Okay, and now those stories are all published, but nevertheless, we have a few million such families in India that provide a fantastic canvas to do genomics. Now that led to the initiation of the Guardian program in 2012, and today from a single Bhai, it has grown to about 300 clinical collaborators from 70 centers with a potential to impact 70 million Indians. How does this program work? Clinicians, when they see patients like Bhai, for whom they have no answers to give, they don't even know what the name of the disease, because it's practically somewhere tucked in some medical textbook that probably, unfortunately, is not taught well in our country, and these patients remain undiagnosed. When they find such cases, they send those cases to us. We do genome sequencing, and when we find the answers, we do a lot of computational work. We have a large supercomputational center in which we do computational work, and if we find a mutation, we go back to the clinician and help the families. But believe me, even with all the skill sets, only about 50% of the cases we are able to find an answer. For the rest 50%, we take up research. Sometimes the research, in a few weeks, we are able to give an answer. Sometimes in a few years, we don't have an answer. So genomics is an evolving puzzle, and it's a fantastic piece of science if you're really like puzzle solving. So what does Guardian represent? It's the largest network on rare disease genomics, not only in India, but in Asia. It has impact in research, education, diagnosis, and policy. It rides on two major technologies, the genomic technology and the digital technology. I'll show you an example of how it is done. And it also gives us an opportunity to work with the government as well as the industry. And so here is I'll give you 3 examples before I move on from this topic. Okay, this family very close to Delhi had petitioned the President of India for euthanasia or mercy killing. Okay, because they had a debilitating neurological condition and there was no name to it. So genomics helped them find the gene that's responsible. In this case, we'll call it the MLC1 gene. It has a pretty long name, as you can see on my screen, but we'll call it, for the sake of ease, the MLC1 gene. And what quickly happened was, as you see in the timeline here, 2016, the case was referred to us through newspapers. We took it to AIMS Delhi with the collaborative help of Professor Shifali Gulati at AIMS. We quickly got to the diagnosis. Again, NGOs like Human Welfare Foundation played key role in supporting the family, and through medicines, through funds, and so on and so forth. By 2018, we had identified the mutation. we had made the publication, and 2019 we had a diagnosis in the market. And in this particular case, the mutation was a very novel mutation. We called it the Nalband mutation in the name of the community, because you won't believe it, there's a second mutation in this gene called the Bansal-Agarwal mutation, and very well documented in literature. So not to confuse with that, we call it the Nalband mutation. And as you can see, we quickly developed a very low cost diagnostics. Remember, genome sequencing still even today costs close to about 50,000 rupees to 80,000 rupees if you do it commercially. And way back in 2016, it used to cost us 2 lakh rupees. That was not affordable to do population level screening. So we really wanted to bring down the cost. So the diagnostic that we have in the market costs us about 1,500 rupees, and it's now available with a leading path lab provider in the country. We have transferred the technology. So by using this low cost diagnostic, we're able to figure out that about 28% of the population actually are carriers of this disease, with 10% being affected. And so that's a huge genetic burden, and those kind of genetic burdens are rarely seen in rare genetic diseases. So you know that with the population size of 50K, you really need low cost diagnostics that we have. You need to go through this community and screen them. And once you have screened them, you could really now start to have multiple advantages. Let me start listing out the advantages. One, okay, the patients are not with an unknown disease, which is, many people think it's a curse. Many people think it's a something bad that they have done in their previous lives that has caused it, and they blame. But they have a molecular diagnosis. They have an end of a medical odyssey. They now have a name to that disease. That's such a important contribution to human emotions. Okay, that's the first thing, a molecular diagnosis. Second, once you have a molecular diagnosis, you can actually do carrier screening. You can find out what is actually ailing that patient or the community at the low cost diagnosis quickly falling in place. Okay, you could do that. Third, you could actually prevent the disease in the next generation by adopting prenatal screening for debilitating diseases. In this particular case, the kids die by the time they reach their early teens. By 13, 14, they actually die. By 8, 9, they become debilitating in their beds, and by 13, 14, they die. So in such conditions, you could now adopt family planning or prenatal diagnosis and prevent the disease. So I call this almost equivalent to the mid 1990s, where you could actually eliminate infectious diseases from happening. We eliminated a number of them by vaccines and many other strategies. Okay, here is an opportunity for genomics to really contribute and eliminate, if not prevent, genetic diseases from the next generation. So I call this a revolution happening right in front of our eyes. Okay, now let me give you one more example. Primary immunodeficiency disorders are disorders where one of your immune cell has gone, is misfunctioning. Therefore, you are susceptible to a lot of infection, and usually the first few years of life, you are in the ICU, and then the baby usually dies off. Okay, really no much treatment available. Okay, so along with Dr. Geeta Govindaradhan at the Government Medical College, Calicut, we really put in a program with Dr. Vinod Skaria, my collaborator. We put in a program for the treatment of this disease and the diagnosis of this disease along with the government of Kerala. Today, we have a clinic that runs for primary immunodeficiency disorders every week. This specialized clinic puts out diagnosis that is supported by the Kerala government in terms of over 30 plus patients. This is slightly older slides, and the numbers should be very high because now they have a full-fledged clinic running that were put on intravenous immunoglobulin supported by the Kerala government. And for patients, after when they became a little bit elder, the patients had access to hematopoietic stem cell transplantation all paid by the Kerala government. So you can see how a single diagnosis, genomic diagnosis, could actually help families access genetic counseling, prenatal screening, and a complete cure to the disease. Now we have the first patients that we had treated actually is entering into the first year MBBS this year. So you can see how much the impact in the society genomics can do. Moving further on, along with the Guardian Consortium and Dr. Radha Mahadevan at Tirunelveli Medical College, we looked at a very unique cohort, a cohort for which there is no name to the disease to begin with. Okay, Dr. Radha Mahadevan identified this adult onset epilepsy. Okay, we eventually ended up collecting 434 patients, which became the world's largest cohort. Okay, for an what I call as an insertion in the SAMD12 gene. Okay, and not only that, we quickly did genome sequencing for 2 patients. Again, couple of lakhs of rupees, and leading to a very cost-effective screening test, which costs as less as 1,000 rupees today. You can screen these patients and family members, and this particular community is about a 10 lakh individuals strong. So you can see there is impact on molecular diagnosis, carrier screening, anticipation, prenatal diagnosis, and you could do a number of other beneficial exercises to the community, and all because genomics could be done not only by the premier medical colleges in India, but this is a medical college in Tirunelveli Medical College is about an hour before you hit Kanyakumari. You can know that even the 3rd tier cities now have access to genomics. Now, all this is because patients came to clinics. Okay. Now, but do we really know what is ailing our Indians? What is ailing in our communities? What is prevalent? Because diseases like the FAMI, the last example that I showed you, or PID, would not be diagnosed by people going to clinics, because these people in many ways don't even have access to clinics. So wouldn't it be better if you knew what really is ailing our communities using genomics as a tool? So that led to us to a program called Indigene, Genomics for Public Health in India, which we ran at in the Institute of Genomics and Integrative Biology at CSIR, along with my colleague Vinod Skaria and many other collaborators. So we asked the question, if you sequence 1,000 self-declared healthy individuals in India, you'd be able to generate some baseline data for what really ails our communities, because technically these are healthy individuals. They don't have a disease, but they would be carriers of diseases that they will come in the later part of life. Okay, so you could really get that information at least a few decades ahead of the disease. You could develop India-centric affordable genetic tests for prevalent genetic diseases and work with industry for scaling up precision medicine applications. And this was also a very unique program because this was a technology demonstrator. This program was done in a flat 6 months time from the sample collection to delivery of the genomics to the nation. And based on this, Government of India actually has launched a number of other programs, such as the Genome India, which we are also part of, that are key person, Dr. Das from Professor Das from IIM just spoke about this program. So you can see how from a single genome, you could go to 1,000 genomes and now have large-scale country-level programs run, because the examples and the benefits are clearly visible. You don't really have to struggle hard to make economic impacts of these kind of data. But those of you are really interested in the economics of how does genomics really help a larger economic macroeconomics, by doing genome sequencing in a patient, okay, you actually save about 50 lakh rupees per person, per family over their lifetime. Now you can just multiply it by 70 million families. Okay, I really had to struggle hard to figure out how many zeros you would add up to that. But remember, that's the impact that you can quickly get if you are able to deploy genomics. And those who are interested, I'm happy to talk. I know all of you are financial gurus. I'm happy to talk about the financial numbers during my question answer session. But that number, remember this number, 50 lakh rupees per family, per individual over a lifetime is the benefit that you derive by deploying genomics at a public health level. Moving on further, what did we learn from this Indigene exercise? That you could really find total about 55 million single nucleotide variations. When I say single nucleotide, that is the amount of data points that we could find people differing between each other from these 1,000 people. And out of these, close to about 18 million were actually novel. That means they are not been reported elsewhere in the world. That means unless we do genomics in our country, okay, nobody's going to come and help us do it. We have to do it ourselves because we are a unique population by and large. And this data is freely available for clinician, scientific community, and for common man in the link that is provided in the screen below. And it's also published now. Oh, I'll give you one more example before I move on, is how does such large programs help the common man? Okay, so cardiac channelopathies is a very complicated word for many of us. But remember, it means nothing that these are conditions that cause something that we all know, sudden cardiac death in the middle years of your life, the productive years of your life. Suddenly you have people who are running to catch a bus, fall flat and die. People who are traveling in trains do not get up the next day morning. You have kids who have exam anxiety and fall flat in the exam hall. We have seen these stories. We have heard these stories. We have heard singers who fell flat right after they walked off the stage. We have heard players fall flat in the field. They all suffer from a disease called sudden cardiac death. And that is caused due to nothing other than in medical literature we call as cardiac channelopathies. That means some ion currents or ion electrical connections in their heart actually misbehaved. Okay, and you'd be surprised if I told you if you condense all our studies from the genomics, that close to about 1+% of Indians actually carry mutations that cause this genetic disease. Okay, now you can imagine with 149 crore Indians out there, 1% is close to about 1.5 crore Indians who actually are walking today, who can, if met the right stress condition or the right trigger, would actually have a sudden cardiac death event that is preventable. Okay, because it's a very, if you knew it, you could change your lifestyle, you could do stress-free jobs, you could not undertake an activity, or if you did, there are beta blockers, there are implantation devices that medicine can help you with today, and there are many such benefits of knowing that you carry a genetic risk of a sudden cardiac death. So you can see how just by doing 1,000 genomes, you are able to arrive at that number. Now imagine doing 1,000,000 genomes. What are the beautiful insights you can arrive at to save human lives? These are all preventable genetic disease. Now this could be really expanded, okay, into newborn screening assays, into prenatal screening, to preemptive screening in adult onset diseases. I've given the top. So if you mind the 1,000 genomes, 1,000+ genomes, these are the top 10 conditions that you listed on my screen. And as in terms of carriers means these are healthy individuals who could carry the predisposition to the disease. That means if both the mother and the father are carriers in many conditions, there's a 25% chance that the child will inherit the disease. And this is a recessive condition. In dominant condition, if you carry the mutation, you will have the disease, like in cardiac channelopathies. These are the top 10 diseases that you have, like sickle cell anemia, thalassemia, you would have heard about. But treatable intellectual diseases, remember the word treatable is important. That means you have a medicine available, and 1 in 100 are carriers in India. And this is treatable. Then in many cases, the medicines are only a few rupees. Wilson's disease is a drug that's available. If you can, it's a preventable condition, neurological condition. If not treated, leads to hepatic transplants or even neuronal very debilitating conditions. You could run national programs, newborn screening programs. You could, for cardiac channelopathies, hypercholesteremia, who have high cholesterols, okay, in their body. You could really run high risk community screening exercises for mitochondrial disease. I think I have an example there, or hereditary cancers. Okay, the requirement for such a number of tests is about 30 million tests annually in our country. Now you can, all of us would now agree upon doing 30 million genomes is not only economically a very distant future task for us. Even in a logistic perspective, we would have to recreate a whole industry. Okay, 30 million tests is a conservative estimate. But of course, now if you could translate and know who carries which disease, you really didn't need to do genomes at scale. You could do focused tests. Okay, because you know who carries which disease, which community carries which disease, and you could run high-risk community screenings. And that's what I'm going to think is one of the way forward for India. For example, mitochondrial genetic diseases in India. Okay, there are about 20 million live births in India. Of this, about 5,000 children are born with mitochondrial disorders. Okay, and these are debilitating disorders in many ways. Okay, so you technically need about 20 million mothers to be tested to figure out who would benefit from this mitochondrial screening, and could they be informed well in advance? Okay, so way back in 2017, we actually transferred a technology to a company called Eurofins, okay, called Mitomatic, and they sold it in a commercial name called MitoSure. They could do three things. It could do clinical diagnosis, prenatal testing, and carrier screening. It was one of the world's first mitochondrial genomic assays for humans. Okay, so again, now the newer version of this assay costs 5,000 rupees. Yes, now you could think of doing assays for 20 million mothers. Okay, now, like I said, we had transferred along with my colleague Vinod Skaria and Mohammad Farooq about 28 tests to a company called Lalpath Lab. Okay, now if you go back and look at many of us get our tests done at Lalpath Labs, a leading diagnostic chain, look at the neurology catalog. Most of it would have come out of our labs. Okay, again, results of our genomic tests now converted into individual gene tests that are about 2,000, 3,000 rupees that are available. Okay, all these came out of the genomic exercises. You could do large-scale genomic screening of individuals, expectant couples. Okay, we transferred a technology to a company called Genomics, which actually offers solutions for expectant couples. Okay, so you can see how genomics could impact. But in a country like India, with 149 crore people, communicating this evidence, communicating evidence is going to be a humongously challenging task. So one of the things that we came up is to piggyback upon the digital revolution that's happening in our country. So we launched something called the Genome Card. Honorable Science and Technology Minister Dr. Harshvardhan, the then Honorable Minister in 2019, just before the pandemic, launched this unique concept where the genomic evidence is actually stored in servers, just equivalent to your funds that you store in banks. But you could still transact using your credit cards very efficiently, isn't it? So why not do that same for genomics? You could store genomics, the genomes in the secure servers, which are privacy protected, but really communicate evidence at a scalable digital platform for communicating the dynamic genomic evidence, because genomic evidence changes by the hour. So we needed a mechanism that is secure, scalable, privacy protected, clinician enabled, and point of care based evidence. So those of you are interested in following up, I'm happy to talk about this, but this technology has been commercialized to a company called Zifo, which is one of the giants in clinical informatics solution providers. It's an Indian company. So I hope we'll all benefit from this technology transfer. Now, moving on ahead, let's say, how did genomics impact SARS-CoV-2? So just as soon as we launched the card, SARS-CoV-2 was just around the corner, and genomics really made a huge impact. And again, India was at the forefront of the genomic technologies. So what CSIR did was launch something called the Mega Lab. Okay, how do you use high throughput genomics for surveillance? And idea was to keep our economic hubs open, okay, by doing this large scale surveillance. Okay, these are not genetic tests. It's not a SARS-CoV-2 RT-PCR. These are genomic tests that tell well in advance what variants are going to come into India, or how do you tackle such variants. So we put together this program along with Illumina, and by July 2020, remember, globally we were the first labs to actually field test this application called COVID-Seq that Illumina had just gotten emergency approval. And when we became the first global lab to do this, of course, we could now leap forward to run this in many of our cities, many of our economic hubs, and we were able to demonstrate how such a genomics approach could really not only sequence singleton SARS-CoV-2 genomes, but actually at scale. And this was quickly adopted. This technology is a very complex one, but believe me, for those of who do the automation, it's very straightforward and high throughput. Really, you can scale up. What had happened was we quickly partnered with 7 states in India to prove that we could do over 2,000 genome sequencing, and all this data is now published. So I'm not going to really go over it, but rather tell you what was the impact of it. The number of states started working with CSIR in this. For example, the state of Kerala really took this surveillance, genomic surveillance, to the core of their decision making, And you can see they had this beautiful maps that way before any of the other states could get, and they actually started using pandemic policy decision based on genomes. For example, here month by month we could go back and show them how genomic evidence in the state assemblies were decided based on the prevailing variants. As we know, the health system in Kerala is far ahead of many of our national or the state labs, and they could really piggyback on genomics. Not only Kerala, actually the state of Maharashtra quickly followed through with Dr. Rajesh Karyakarte and his team really chipping in to do a large-scale genomic surveillance for that district-wide surveillance to know what was ailing their state and how to use that for issuing lockdown statements or removing lockdowns, and how people move from one place to another. A lot of pandemic policy decisions were based on genomes. And very quickly in March 2021, the government of India, through funding from Ministry of Health and Family Welfare, DBT, CSIR, ICMR, and the state governments, put together this national program called INSACOG, which today is a very strong program run by DBT and many of its partner science institutions to really map out what really is the SARS-CoV-2 variant in our country and such. So I can show you how from a single lab that showed a proof of concept in June 2020 as the world's first concept to running a national program to actually we have run an international program. I'll just come into a minute. But before I do that, let me tell you what is the advantage of doing a genomic surveillance. Okay, many of you would have asked yourself that question. Why are people wasting money on this? Actually, there's a huge advantage because here is the Delta waves. Many of us vividly remember the Delta wave and how devastating it was in our country. Okay, the first reminiscence that we had Delta was in August 2020. The wave actually happened on April 2021. Okay, and you can see that was the first time we noticed something was very different floating around in our population from Maharashtra. And remember, then there was this nice lean period. We actually stopped doing genomic surveillance. We should not have stopped. We should have continued. We would have picked up more of these and taken precautions. And that was when the first Yavatmal and the Amravati cases started showing up, and then the massive wave that came in. So, you have a lead time of about 8-9 months if you really deployed genomic surveillance at scale and followed it to the last dot. And that's what genomic surveillance gives you, a lead time. But what we did quickly was scale this up along with WHO for the CRO countries, which is about 11 countries now. And you are able to really develop a rapid genome sequencing protocol, which in about 24 hours time actually gives you reports on clusters of information, cluster deaths, time critical events. You have a plane load of people sitting at the airport. You could quickly deploy genomics at the point. And this is something that WHO now takes up for all the CRO countries. Again, a technology that originated right in India at IGIB, now being deployed at international scale. So what I think in terms of COVID impact, if I look back for the last 2 years, I think there will be 1 million direct beneficiaries. And I think there is at least a 10x proportion of indirect beneficiaries that would have come up through various programs that I've talked about. Of course, there were kits that we made that were genome guided, which I have not really included in the talk today. But those are also the impact that came out of this program. And we actually knew how to work with. We bridged a very important gap. That gap is science, technology, clinical was an axis that was always working. But we actually went from science, technology, clinician to policy making. And that was the gap that I think was a significant gap that we really took a leap into helping policy decisions and working with administrators. And now you ask the question, with all this knowledge that we have generated, can we really hit the road running? Okay, and that test case became the lumpy skin disease. Okay, for those of you who are new to this, it's a disease in the cattle that is really causing a pandemic in 15 states in India with over 100,000 cattle deaths. Okay, with 2 million cattle affected, and cattle that get affected are in a debilitating condition. Their milk production goes down, and in a very India is in the threshold, probably in a very serious economic turning point in terms of our agriculture and bio or dairy product segment. Okay, so with this kind of a disease going on in our communities, could genomics help? So from sample to actually putting solutions on the ground, we only took 1 month. We didn't take 2 years like we did in COVID, and not definitely a few years that we did in the humans. 1 month. Okay, we had a genomic surveillance that is called SnapSeq, ready, done, published, and proof of concept done and transferred to a state government. More RT-PCR done, already transferred to a state government. The point of care, we are in the process of transferring to state governments for deploying. And today we had the Ministry of Health in the Animal Welfare Department walk up to us and saying, tell us, how do you take this forward? Okay, so you know that things can be done if you have solutions, and genomics does provide solutions in real time. This is my last slide before I say, so what have we done in terms of genomic impacts in India? Okay, we not only do population genomes, we actually do pathogen surveillance, we do rare disease genomics. We also provide about 300 plus genetic tests free of cost to needy patients right out of IGIB and the CSIR labs. We work with clinicians to teach them genomics. We work with industry to translate them to commercial deployments. In this whole process, we actually do what we really are passionate about, is science and research. And we have not only worked in India, we have both national and global partners whom we work with, and we are really proud about how a single technology could impact such a large number of people. I think in a relatively conservative estimate, we could really impact close to about 50 crore individuals today if you deploy genomics at scale in this country, and these are direct beneficiaries. The indirect beneficiary probably would be a larger number. Those of you are interested, genomics is not, we don't do genomics just for the sake of genomics. We do it for education, diagnosis, policy implementation, and we have captured all this in this large review that we published about a year and a half ago with our experience. You are welcome to write to me. I'll be happy to share this. It's also available in public domain. I'll acknowledge a large number of people who have made this possible in the COVID genome surveillance programs. A lot of state government hospitals, state government health facilities, and organizations. Of course, my own team with Vinod and Rahul and Paras, who have been the bedrock into taking this forward. In terms of the human genomes, a lot of clinicians, collaborating centers, scientists, and large number of students, and of course, very sustained funding and dedicated funding from Council for Scientific and Industrial Research over the last decade and a half. And that is something that I think is very important. Not all funding agencies really believe in such sciences and keep funding you for a decade and a half to see the results. And I'm very proud to say that CSIR is one such organization, and I'm happy to be part of it. And shown on the screen is our lab. If you're ever in Delhi, please do stop by. I'm happy to give you a tour. I'll stop there and take questions. Thank you. Back to you, Dr. Das, Professor Das.
Thanks, Professor Sridhar, for your very well thought, and this talk provides a very good understanding about the current progress in this area, and also how genomics is now integral to any disease research and drug discovery. So on behalf of FIDR, IIM Lucknow and XIM, we are very much thankful for this enlightening speech talk, and we'll be very much interested for this kind of discussion in future, some kind of panel discussion, more about policy related issues, so that this can, very few people are working on this area, how that can be practiced in more hospitals, and we can popularize this particular discipline. And looking forward more elaborative discussion in future. On behalf of FIDR, IIM Lucknow and XIM, I again I'm thankful to you for this very thoughtful and interesting speech. Actually, this kind of our students, community people, they are very much interested to do some kind of discussion in our institute also. We have discussed, we will be inviting you in future for this thing. Thank you very much.
Thank you, sir. Thank you. for those nice words. I'd be happy to contribute to such interactions.
Thank you very much.
Yeah, I see a number of comments and questions.
Yeah. We
If
can,
there
sorry,
is
we
time,
can
we
take
could
up.
take it. I'll let you decide, sir.
Yeah, we can take up some questions. Yeah, sorry.
So should I go ahead then?
Yeah, please.
Yeah. So there's a question on how can we bring genomics to the forefront of policymakers across sectors, human lives, animals, and agriculture, and so on and so forth. I think it is through forums such as this and through government, talking to government agencies, think tanks, and writing policy documents is something that we could do that. And I've given some examples of this in my talk, but I'm happy to discuss more on this. The next one is about why genomics is thought to be expensive and difficult to get to common people. So yes, genomics was expensive and difficult in terms of technological know-how, but like I said, COVID was a real eye-opener. Okay, the COVID genomics was offered at 2,800 rupees per genome. Okay, at scale, it could be brought down to even something close to 1,500 rupees. And one of the thought process, like the lumpy skin disease assays, are close to that price point. And I showed you the human genome assays, again, are all within 5,000 rupees. I think the solutions that work for India are very different from the solutions that have been propagated by Western countries, and we need to rethink about this solution, and that's something that we are very passionate and doing. Professor Patnaik, if you are interested, I can give you very specific examples. Do write to me on how to bring down the cost. There is one more question on is there something done for Northeast state in terms of genomics by Professor Srinivas. Professor Srinivas, you are from Aizawl, and actually I have visited Aizawl twice. Okay, the cancer is a major issue there. So one of the first tests, we call it the genomics-based pharmacogenomic test for a drug called as 5-fluorouracil, which is given for solid tumors. And I know Northeast has a large proportion of cancer cases. Okay, this was genomically derived. The mutation, the variants identified that are very popular in our population, and those have been transferred to the university, central university at Mizoram, Aizawl, to Professor Sendil's lab, who now offers it to the hospital, the Cancer Research Hospital, from his lab. So you can see even the farthest flung regions in Northeast have benefited from genomics. So we just need to keep expanding this network. I think there is one more last question. How does genomics help livelihoods of communities with agriculture? While there are a number of examples in agriculture which I have probably not listed, I'll go back to the lumpy skin, which has a direct impact on the bioagri economy. You can see how genomic surveillance can identify cattle, make sure the vaccination is given to the right cattle, put policies in place to prevent the strategy. You've seen that how it happens in COVID. I'm pretty sure it will happen in other infected diseases also. I'll stop there and take any other questions that you might have verbally. Thank you.
Yes, sir. So there's one more question mentioned in the chat.
Can you just read it out? Maybe I missed it.
Yes, sir. Yes, sir. I can read out the question for you. So the question Anshuman Nanda sir has asked, as a policy expert, how can we bring genomics to the forefront of policymakers across sectors, human lives, animal lives, agriculture, and so on and so forth?
I think I answered this question, but I'll repeat it once again. So throughout my talk, you heard about how economic benefits can be derived just by monetizing on assays, monetizing on finding a molecular diagnosis. And once you know the molecular diagnosis, you could devise strategies for not only looking at who carries this disease, actually prevent it in the next generation in terms of human lives. In terms of SARS-CoV-2, we could actually use that to really find out what variants are there in which communities, which states, and actually example was that Kerala could actually devise pandemic policies. They could open up the airports and lock up the land borders very strictly, just because they realized that the infection was actually coming from the land borders and not from the international travelers. So there were policy decisions that could be gained out of genomic evidences. And there are many such examples. I'm happy to talk beyond this meeting.
Yeah. So this is Anshuman. I actually had that question. So
Yes,
my question is a little more pointed towards the policy angle. That is, Kerala has already taken a major lead as compared to other states. So I would like to understand what exactly were the initial actions by the think tank as well as the government. What were the initial pivotal key activities which actually led to this advantageous position vis-a-vis other states?
So both Kerala, Maharashtra, and now practically every state in India through the INSACOG program does participate in the genomic surveillance. But now going back and asking what initiated this in Kerala was largely because they knew that they were going to get an influx of people coming in from other nations, because Kerala, large population of Kerala lives abroad. So they were scared that they're going to import in a lot of new varieties of the SARS-CoV-2 variants. So it just so happened that the Principal Secretary Health was also a clinician. So he had been following our work from the primary immunodeficiency disorders which we had done in Kerala at Calicut Medical College. So he had been following that work. So there was some pretty previous work that we had been introduced genomics to them. So when he first noticed this genomic assay that we had done with the 7 states, he quickly jumped up and say, why could we not use this to find out if Kerala is getting the variants from international travelers, or is it local local spreads? So those kind of questions, when they wrote down, they looked around to see that only genomics could provide those answers. Second, if you also remember very nicely, Kerala had a very long persistent COVID infection going on, and that was in the news for quite some time. And whereas their mortality rate was very, very less compared to the national average. Okay, so and they could do this primarily because they had genomic evidence to play with. So again, having the right people who have experienced genomics or tested the power of genomics, having clinicians also advocating, having policymakers as clinicians turned policymakers really helped us bridge that gap. In Maharashtra, though, it was a very different set of equations. Maharashtra was very scared about the Mumbai outbreak, and therefore they wanted to quickly have all tools in their disposal, and that led to Professor Karyakarte and the previous health secretary at Maharashtra take up genomics as one of their evidences of the many things that they did. Of course, in the meantime, the world had really shown that how genomic evidence could contribute to preventing or the spread of SARS-CoV-2, and that led to the national program by and large.
So to summarize it in 2 lines, wisdom at the right level and a sense of impending danger. Actually, these are the 2 fundamental reasons which led to Kerala and Maharashtra's quick catch up into the.
So I think wisdom at the right level is a beautifully coined, but I also say some bit of brush with genomics prior to COVID. Okay. Because they knew what the power of genomics was from the primary immunodeficiency work that we had done. And that really helped them have the confidence to take the next step.
Thank you, sir. I got the thank you.
Any other questions? Please feel free to ask. If not, back to you, Professor Das and the host.
Yeah. So I think there is no other question. We can. Would you like to speak something, FIDR? So we can now,
Yes,
yeah.
yes, Professor. My turn to give a vote of thanks and some on the discussion. Yes, Professor Sridhar Sivasubbu, it's a big thank, and it's a great pleasure to listen to a public intellectual and a scientist on genomics, and that public health needs today in India the most, because public health is very critical at this in a critical juncture. It's not necessarily post-COVID, but prior to COVID too, and it is a overwhelming response. And you rightly said the discussion and how scientists, clinicians, and practitioners and policymakers collaborating and contributing to this. It is always great pleasure to have experts and participants from more than 6 states, as well as in nationally as well as international participation for a August gathering like this. But these scientific deliberations needs to be customized in the sense of policy responses more accurately. I am so happy to share that by calling to now, you have got INSACOG network. So that's the landmark of the journey, and many congratulations for that. And genomics is no more a scientific discovery only. It is a public health reality for its practical interventions. And you rightly said that it has touched the National Health Mission and many more states to join. I recollect often Prime Minister's speech, like what scientists are doing and world must listen. And this is a true sense, a road railing exercise and achievement in the landmark in the true sense, India to be called as a global leader in the discovery of genomics. And I congratulate even World Health Organization and the Southeast Asia Regional Office to scaling it off. And I am confirming that public health is no more in danger with this sort of discovery. And I, on behalf of being first public health advisor to National Rural Health Mission, I'm quite optimistic and quite happy to express that this strengthening of health system through achieving universal health coverage by 2030 is going to be no more, it will be a distant reality. It is going to happen very soon with this sort of discovery, which needs further deliberations and customizations along with the states as well as its parties, especially the students of interdisciplinary research. And as a genomics expert, public health is my cup of tea. I always welcome any sort of complementarity in the field of research, because research brings out evidence to feed the policymaker and a successful public policy and public health policy. That is what precisely you brought out in this very brief presentation. And I, on behalf of School of Governance and Public Affairs, Javier University, XIM University, Bhubaneswar, would like to thank FIDR for this initiative, which is very timely and which is very much appropriate in a time when we repeatedly say public health is in critical juncture. So, and also the Center for Environmental Education, Indian Institute of Management, Lucknow, if I am correct, and Center for Environmental, CMEE, IIM Lucknow, as a premier institute of India for their collaborations, and all of you who have participated today, a heartfelt gratitude to all of you for their question, and Professor Sridhar Sivasubbu for clarifying each of these questions and giving an opportunity to interact further in future. The Think Hatchery Talk series has been attracting people from all over the country and other countries as well. And I wish all of us have a happy season ahead and Happy New Year 2023. unless we meet again, and that's what I would like to mention here. And over to FIDR.
Now, wonderful session has come to close. Thank you all for your enthusiasm and participation.
Thank you, everybody. Thank you for the kind
Thank you.
words. Thank you.
I would request Dr. Sridhar to make me the host.
Did I? Okay.
Dr. Sridhar, thank you. Dr. Paningai, Dr.
Thank you.
Das, Professor Das.
Thank you so much.
Thank you much.
Thank you, Dr. Sridhar. Thank you, everyone.
Sure. Thank you. Thank you. Thank you,
Really
everyone.
good.
Okay. I've made you the host. Thank you, everyone, once again. And I thank you for the invitation. I enjoyed the interactions. Namaste.
Namaste.